Hello, this is Dr. James Camp at Lee College in Baytown, Texas, and this is my introduction to anatomy and physiology, sometimes called the twin sciences, though as you'll see, they are quite non-identical twins. How do we know the difference between anatomy and physiology? How do we know what each one studies? Well, oftentimes people say anatomy studies structures and physiology studies functions, but let's see to what extent that's true. Let's start with the idea of what the words themselves can tell us. Ana is Greek for moving apart or up, and tomia is Greek for the cutting action. So anatomy, in other words, means cutting apart. In this particular case, it means cutting apart of the body. So we can tell that it's a science of body parts, of what you get when you cut apart the body. On the other side, physis means nature, and logos is a Greek word that has lots of meanings, including words, thoughts, philosophies, both words and the thoughts that the words express. So we know that physiology, physis logos, is thoughts about nature. So this is the science of natural things. I thought that was biology. Well, okay, we need a better definition. What does Webster's dictionary say? Webster says anatomy is a branch of structural biology that deals with the structure of organisms. Okay, so we're back to that idea, anatomy studies structure. It's the science of body structures. Physiology, Webster says, is a branch of biology that deals with the functions and activities of life or of living matter and of the physical and chemical phenomena involved. So again, we're back to this idea of functions. Physiology then is the science of body functions. So what do anatomists and physiologists really do? Anatomists study the overall markings and regions of the body, while physiologists study the overall functions of the body as a whole. That much is probably pretty obvious. Anatomists study locations, shapes, and structures of individual organs. So where is the heart found, and what is its overall shape, and what parts does it have? Physiologists study the detailed functions of each body system and individual organ. So they would be more, a physiologist would be more interested in what the heart does in its location than what its particular shapes and structures are. Anatomists study the tissue patterns and cell structures of each organ. Physiologists study the chemical and cellular details, mechanisms that explain each function. So here we're getting down to the nitty-gritty of what we'll really study in physiology for a lot of these systems in this class is the actual chemical or cellular explanations for how a muscle contracts or a nerve fiber sends a signal. And finally, anatomists study how each structure relates to its function. This is a science called functional anatomy. A lot of people think that as soon as you start talking about functions, you're in physiology, but that's not really true. In order to understand the biceps muscle of the arm, for example, you've got to know what its function is, which is to flex the arm, or to flex the forearm, really. Physiologists, on the other hand, study how each function helps the body maintain homeostasis or respond to challenges, or maybe both. Now, are anatomy and physiology completely separate sciences? Of course not. We just talked about the fact that functional anatomy is really kind of a branch of anatomy, even though it's talking about functions. Well, physiology and anatomy are considered twin sciences because really the anatomy and the physiology of most body parts are linked. In order to do a function, you need to have a structure. And in order to understand the structure, you need to know its function. Why? Well, there's a common saying, form follows function. Form or structure follows function, what it does. This can be clearly seen when we study the bones later in anatomy, where we're going to see that the bones are not perfectly smooth. They're not like a little dog bone that you buy at the store. They contain bumps, depressions, roundings, holes. And to understand each of these things really requires some knowledge of the fact that each one of these protrusions or depressions on the bones serves a function. Okay? Bumps are often attachment points for ligaments or tendons. Depressions may be a hollow space for another organ to fit in. Rounded areas are often part of a joint that needs to move smoothly. And finally, holes or notches are almost always there for something to move through them, whether it be a blood vessel or a nerve or a tendon or ligament or muscle. But there's a caveat to this. Not every form has a present-day function. Can you think of some part of the body that doesn't seem to do anything useful? The issue here is that evolution is very good at adding new features that we need. Okay? If a human being needs to walk on two legs in order to survive, then we'll develop the structures we need to do that. If humans need a bigger prefrontal cortex in our brains to socialize with other humans, we'll develop that. And if we don't, we'll probably not survive, or even if we do survive, we won't reproduce successfully. But evolution is pretty lazy about removing features that we no longer need. A classic example of this is that we have this perfectly useless tailbone called the coccyx. Do we have a tail anymore? No, but we have a tailbone. In fact, our embryos grow a complete tail and then reabsorb it into the embryo and just leave the tailbone. But the tailbone has stuck around with us for millions of years after our species gave up having a tail. Now, I don't know about you, but I think it would actually be kind of cool to have a tail. I mean, have my tail hold my grocery bags while I open the car or something like that. Okay. But I don't have a tail, but I still have a tailbone. Note also that not every form is a perfect fit for its function. Evolution often works by editing features we already have to work just well enough rather than adding new features that work perfectly. A classic example of this, still staying in the skeleton, is the human spine, which is based really on a quadruped design. Our spines are not very different from the spines of animals that walk on all fours. While our spine has adapted to work well enough at keeping us on 2 feet, how many people do you know that have back problems? It's a design that has some problems. Okay, so now that we understand what anatomy is and what physiology is and the link between the two, let's take a little detour to talk about the types of things that anatomists might study and the types of things that physiologists might study. Anatomists might study gross anatomy, which not ooh yuck gross like a 5th grader would say, but gross as in the idea of something gross is big. Okay, so gross anatomy is the study of structures large enough to be visible to the eye. Microscopic anatomy, on the other hand, is the study of structures small enough that you need a microscope to see them. What kinds of things do we study in gross anatomy? Well, we might study the surface of the body, surface markings and surface landmarks and body regions. We've already done some of that in this class. We might study regional anatomy, where we say, well, what are the structures that make up the head? Well, it's got eyes and ears and a nasal cavity and an oral cavity and a cranial cavity. And how do these things interact with each other? How do the cranial nerves cross from the cranial cavity to the face, and so forth and so on. Or we might study sectional anatomy. This was once upon a time purely an academic discipline. If you have a person's head and you slice it like this, or if you have a person's body part and you slice it like this, what are the parts that you see? How does that help you understand how everything relates to each other? But in an age where we have MRIs and CT scans and all of these things that take virtual cross sections through somebody's body, sectional anatomy has suddenly become a really applied field. Systemic anatomy is kind of the sort of anatomy we'll be practicing in this class, where you split the body up into a bunch of systems and you study the anatomy of an entire system at one time. And finally, organ anatomy. Pretty much any of the approaches that you take, whether it's regional, sectional, or systemic, at some point you're going to get down to trying to figure out which organ you're looking at and what the structures are within a particular organ. Microscopic anatomy, we've also got a few options. We can dive all the way down to the cell level and study cytology. This is the study of the shapes and structures of individual cells, and sometimes even the things inside those cells, the organelles within the cell. Histology is the study of tissues. Tissues are made up of multiple cells and also several other components like protein fibers. So histology is using a microscope to understand what tissues make up an organ, how they're arranged perhaps into layers or into regions. And then that brings us up to the microscopic version of sectional anatomy, which is to say, whenever you're looking at a microscope slide of an organ, you're looking at an organ that has been sectioned into a very thin slice that can be put on a microscope. And so ultimately, all microanatomy becomes sectional anatomy, at least the way we approach it in this class. Okay. We now understand that there are lots of ways to approach anatomy. How about physiology? Well, physiology is not organized into subfields the same way as anatomy. We don't have gross physiology and microphysiology. We don't have regional physiology versus, sectional physiology. We have physiology. Some physiologists are going to focus on a topic. A physiologist might study, for example, the effect of a new blood pressure medication. But blood pressure affects not just the heart, but also the kidneys. And the heart and kidneys have effects on blood pressure. So trying to study blood pressure requires studying one effect across multiple systems. Or someone might be interested in the causes of osteoporosis in postmenopausal women. Calcium in bones certainly seems like a skeletal system problem, right? But menopause involves hormones, so that's endocrine. But the hormones come from the reproductive system. And the calcium that is being removed from the bones also involves the kidneys and the digestive system to replace that calcium. And there are a number of systems that get involved in studying this one simple problem. Other physiologists, on the other hand, study all the functions and disorders of one particular organ system. These are people who you tend to find in clinical practice. You'll find a cardiologist or a neurologist or a gastroenterologist. Most doctors who specialize in one field of what we would call systemic physiology. Some physiologists are going to study the gross effects of organ function on the organ or the body as a whole. So, for example, a sports and exercise physiologist might study how muscle fatigue develops and how to prevent it at a whole organ level. An endocrinologist might be interested in how a particular hormone affects its list of target organs, and for how long, and how you would diagnose that, how you would understand the symptoms, et cetera, et cetera. Other physiologists, the ones who are more involved in academic research, I suppose, zoom in on the cellular or biochemical mechanisms that explain a function. So they're not interested in whole muscle fatigue. They're interested in the nitty-gritty details of how a molecular motor within the muscle fiber causes the muscle to get shorter, to contract. They're not interested in the effect of the hormone on the whole body. They're interested in the effect of the hormone within an individual cell. Okay, we'll talk about physiology at both of these levels throughout this class. We'll talk about the gross effects of muscle fatigue and of hormone actions on the body, but we'll also get into these ideas of the cellular and biochemical mechanisms, because you can't understand modern medicine and the drugs that we use and the way they work without some understanding of what's going on inside your cells with all of these interactions. But all physiologists do have kind of a common toolkit in terms of the questions they ask, whether they're looking at one system or all the systems, whether they're looking at gross physiology or microphysiology. They might ask, how is homeostasis of something, calcium, blood pressure, maintained? How does the bone respond to changes in calcium? How does the heart respond to changes in blood pressure? How does the heart respond to changes in the exercise demand of your body? Those are other, less homeostatic changes. They also want to know another pair of questions. How is a particular activity controlled? How do we know when to contract a muscle and when not to? How do we know when to speed up the heart rate and when to slow it down? And how does that organ get the energy it needs to function? What feeds a hungry muscle or a hungry nerve? And finally, there are physiologists who almost all physiologists at some point ask the question, well, how does my body part get sick? What is the pathophysiology of the heart or the bones or any of these other organs? Okay, I want to zoom in on those first two questions for the rest of this presentation. First, homeostasis. Homeostasis is known as the body's active mode most of the time. Most of the time, you're not in a state of crisis. Your body is trying to maintain a stable environment. But sometimes the body's active mode changes because you're facing a demand. You're sick and you're trying to fight off an invading organism, a microorganism, or you're exercising and you need more oxygen and blood to your tissues. These are not extraordinary changes, but they're not homeostasis anymore. We've disrupted homeostasis to respond to a challenge. One of the issues I have with a lot of the textbooks out there is that they say that the entirety of physiology is about maintaining homeostasis. And yet, in fact, some of them even say that disease can be defined as moving away from homeostasis. But would you then say that increasing your heart rate while you exercise is disease? No, of course you wouldn't. So clearly, the body really has two different modes in a way. It's got a homeostatic mode and it's got a responsive mode. Homeostasis is going to maintain internal variables. And whenever I say the word variable here, I'm talking about anything you can measure that's capable of changing over time. Your heart rate, your blood sugar, your body temperature, your blood oxygen, any of these things are variables because they vary over time. And homeostasis is going to try to keep those variables within normal limits, okay, to keep the body stable and healthy. But the response mode is going to say, you know what, for a short time, I'm going to set this variable outside its limits to meet a very specific demand. The body needs more oxygen, so I'm going to increase my heart rate. The body needs to fight off an illness, so I'm going to raise my body temperature a little bit. Homeostasis is what defines healthy values for a resting body. We try to get homeostatic values for patients when we do a lab test, for example, because that's the healthy value, at least most of the time. But again, the response mode is capable of altering those values for a short period of time, and that can still be healthy. Okay, a heartbeat of 100 beats per minute is considered tachycardia if you're at rest. But a heartbeat of 120 beats per minute is considered fine if you're running on a treadmill. So a couple of examples here. I've already mentioned these examples, but I want to spell them out. In homeostasis, the heart rate is 70 to 80 beats per minute. 100 would be considered abnormally high. But in response to stress or exercise, the heart rate can easily double that, 140 to 160 beats per minute, and still be considered within the healthy range. In fact, most exercise scientists say that that's what you want for a good cardiovascular workout. When you're in homeostasis, your body temperature is about 37 degrees centigrade, or 98 to 99 degrees Fahrenheit. In response to an infection, however, the body temperature can rise slightly and still be healthy. Most doctors at this point won't consider you to have an unhealthy fever until you're up about, above 101 degrees, depending on the patient. In homeostasis, blood sugar falls within certain limits. We all know that. We've heard about diabetes as hyperglycemia, too much blood sugar. But did you know that under stress, certain hormones in your body can cause your blood sugar to deliberately rise for a short period of time, or even for a long period of time, to enable your body to withstand that stress? If you're running from a bear in the woods, that kind of stress requires some extra blood sugar. All right, so now for the rest of this presentation, I want to talk about how homeostatic and responsive processes are controlled in the body. It all usually starts with the change in a variable. When a variable changes, this is called a stimulus. For example, blood oxygen falling during exercise or blood sugar rising during a meal. The stimulus is going to be detected by a receptor somewhere. The receptor might be a designated nervous system receptor, like a chemoreceptor embedded in a blood vessel that detects the blood oxygen. Or it might be a specific organ that's programmed to detect that stimulus, like the islets of the pancreas, which detect blood sugar. Then a control center, a second part of this response, decides how to respond. The chemoreceptors in the blood vessels are going to signal to the respiratory center in your brainstem. The islets of the pancreas, on the other hand, are both the receptor and the control center. Sometimes those two functionalities are put together in the same organ. Once the control signal center decides how to respond, it sends a signal to an effector organ. Nerve signals, for example, will go to the breathing muscles to adjust your breathing rate to fix the oxygen problem. The hormone insulin will be sent by the pancreas to a number of target organs to fix the glucose problem, the blood sugar problem. Finally, some sort of response is going to occur, or there wouldn't be any point in doing this. The respiratory rate increases, making more fresh oxygen available, or sugar is removed from the blood and stored in various organs. So if you take out the middle steps, what you can see is that a variable changed, that was the stimulus, and a response occurred. Notice, however, that in both cases, the response fixes the problem that created the stimulus, or at least it addresses the variable change that started with the stimulus. When the response somehow affects, or we say feeds back on, the stimulus, we've created a feedback loop. Negative feedback loops are going to reduce or reverse the change that caused the stimulus. If blood oxygen falls, we're going to make more oxygen available and raise the blood oxygen. If blood sugar rises, we're going to store the extra sugar somewhere and reduce the blood sugar. So we're going to return things to normal or restore homeostasis. Most feedback loops work that way. Positive feedback loops are kind of rare. They actually increase the change that caused the stimulus. In other words, they feed back positively. If there was a little change, they feed back to make it a bigger change, and then a bigger change, and then a bigger change. Okay, to put it another way, with a negative feedback loop, if there's an increase, we're going to feed back negatively to decrease it. If there's a decrease, we're going to feed back negatively to increase it. We're always going to push it back to where it started. With a positive feedback loop, there's a little increase and then a bigger one and a bigger one and a bigger one because we keep feeding back positively on the change. This is those 6 pieces of a feedback loop that we just talked about in text form. This is those presented in a loop shape, okay, in a loop diagram. Let's look at a negative feedback loop. And since we don't know a lot about the body yet, we're just starting anatomy and physiology, I'm going to take a non-body example. I'm going to talk about what happens when it gets cold outside. Okay, so the stimulus is that your house temperature is going to fall because it got cold outside. Where's the receptor? The receptor is a thermosensor somewhere in your house, usually built into the control center, which is called a thermostat. Usually these two work as a single unit. They're built, in fact, as a single unit. But your thermostat wouldn't be very useful if it didn't have a thermosensor, a thermometer, built in somehow. Okay, so the thermosensor picks up a drop in temperature and informs the thermostat. The thermostat decides, well, we've got to raise the temperature. So it sends a signal called electric power to an effector. What's an effector? An effector is anything that can cause an effect. So that can make an effect happen. Put it a better way, it's anything that can affect the system we're trying to fix. So what can fix what can affect a cold house? A heater. Okay, so somewhere in your house you have a heater. Maybe it's a furnace built into your attic that blows cold air, blows hot air into your house. Maybe it's radiators built into various parts of your house that radiate heat that way. Maybe you have a space heater somewhere. However it's designed, you've got something that creates heat. Its response is going to be to warm the air in your house, okay, so that your house temperature increases. Notice that response feeds back on the stimulus. It feeds back in a negative way because the house temperature increasing is the opposite or the negative of the house temperature decreasing. So the response is equal and opposite to the stimulus. That fixes the problem and ends the feedback loop. A few examples of negative feedback in the body. Let's say your body temperature increases. You start to sweat. You dilate your dermal blood vessels. All those things lead to your body temperature decreasing and you return to normal. Let's say your blood sugar increases. Sugar is removed from blood. We just did that example. It's stored in various organs, and so the blood sugar decreases and returns to normal. But it's not always an increase-decrease pattern. It can work the other direction. Let's say blood calcium decreases. Your body needs calcium to function, so calcium is released from your bones and the blood calcium goes back to normal. Or let's say your blood pressure decreases. If you don't have enough blood pressure, your blood can't flow, so your blood vessels are going to constrict to squeeze on that blood and raise your blood pressure. The blood pressure increases and you return to normal. Notice how all of these examples act to maintain homeostasis. Negative feedback is almost always used to maintain a homeostatic environment, and it's almost always used in homeostatic regulation systems. Now let's look at a positive feedback loop. For this, we do really need to look at the body as an example because I can't come up with any really easy to explain positive feedback loops outside the body. So let's take an example of a woman who is in labor. The stimulus is, in this case, is that the baby starts to enter the birth canal. Okay, so there's a receptor. There are stretch receptors built into the cervix and the vagina that detect the baby moving into the birth canal, and they send a signal to a control center. The control center in this case is the hypothalamus of your brain, which happens to be your body's main nervous system, endocrine system link. Well, the hypothalamus is going to send a signal, and the signal it's going to send is the hormone oxytocin. Some of y'all know its artificial cousin, pitocin, which is used to induce labor. Oxytocin is going to go to an effector organ. Well, what kind of organ can affect a baby entering the birth canal? Well, the uterine muscles can have the effect we're looking for. Now, here we have to ask ourselves, do we want negative feedback? Negative feedback on a baby leaving the womb would be push the baby back in. That's not what we want. We want the baby to be born. So we actually need positive feedback here. We need something that will push the baby further into the birth canal. What would do that? It would be the uterine muscles increasing their rate of contraction. Okay, so we increase our contractions, and that's going to push the baby further out. Well, we started with the baby, it's pushing the baby out of the uterus, so I should say it's going to push the baby further into the birth canal. Okay, so the baby moving into the birth canal is exactly what we started with. And so this effect is positive feedback. So we've now got the baby entering the birth canal even more than it was before. So this arrow gets exaggerated. More stretch receptors are activated, so that arrow gets exaggerated. The hypothalamus is now even more aware of what's going on, so it's going to release more oxytocin. That oxytocin is going to go to the effector, and the effector is going to strengthen its response. So we'll get even more contractions, pushing the baby even further into the birth canal, so we get more positive feedback. Can you see how this system works? Every time you go through the loop, the whole system just gets stronger and bigger, and the whole system gets stronger and the effects get bigger. And this system will continue. It will build up and build up and build up and go on literally forever until something breaks the cycle. Positive feedback loops, unlike negative feedback loops, positive feedback loops need what we might call a stop condition. So at some point, we reach a stop condition. What's the stop condition for giving birth? The baby being out of the body. Okay, so once the baby's out, then there are no more stretch receptors sending signals to the hypothalamus, so the hypothalamus doesn't release any more oxytocin, so the uterine muscles will slowly wind down their contractions, and the labor will come to an end. Some more examples of positive feedback, most of which seem to involve the reproductive system. When a baby suckles at its mother's breast, the breast is stimulated to eject, we say, or let down in common language, milk from its glands into the nipple where the baby can get it. As a result, the baby suckles more. Okay, there's milk here, let me get it. That's going to stimulate more milk to be let down, and this process continues until we reach a stop condition. The baby stops nursing, or the mother runs out of milk. When platelets stick to a broken blood vessel wall, okay, your blood vessel is broken, you're bleeding, you need it to stop, right? The platelets stick to the broken blood vessel wall, they release factors that attract more platelets. So more platelets stick. They release more factors that recruit more platelets. So more platelets stick. So more factors. So more platelets. So more factors. So more platelets. And that process continues until we've completely covered the wound and we have what's called a platelet plug. Then other factors will start that form a fibrin mesh, and that's also a positive feedback process. So the entire process of what we call homeostasis, stopping the bleeding, is built around positive feedback. Why? Because we want to speed up, we want that to happen as quickly as possible. Finally, a little more complicated example, one we'll get to at the very end of this class. When the female reproductive system wants to ovulate, a hormone called follicle-stimulating hormone causes the ovarian follicle to grow. The growing follicle releases more of a hormone called estrogen. Estrogen has this effect of causing more FSH to be released. Well, FSH causes the follicle to grow even more, and the process continues over and over and over again. Note that these examples are largely not about homeostasis. They're about accomplishing some sort of a task. Feeding an infant, stopping the bleeding, preparing for ovulation. We're trying to do something and get it done quickly, usually. Okay, that's the end. You now know the basic approaches that anatomy and physiology take to studying the body, and you know how, with the form follows function motif, we're going to try to integrate those two into the study of both in my class. And you know a little bit about negative and positive feedback. Stay tuned. We'll talk about feedback loops throughout the rest of this class. Thank you for your attention. Good night.